Hi guys, welcome back. I am Heidi Histrick and this is part 3 of the Carolina Superbugs Lab. I've already made videos about how to prep and pour your plates for the lab and how to make your broth for the lab. If you haven't watched those, the links are down below. This video is going to show you how to prepare your 4 conformation plates. So that's the next part of the lab. But with any microbiology lab, our first step is always to sterilize our area. So I'm going to be using my 10% bleach solution to spray and wipe it down. That's because we're trying to grow pure cultures, so we don't want any bacteria, fungus that's on the surface to potentially contaminate our bacterial cultures. My area is sterilized and I have gathered materials that I'm going to need for my conformation plates. I'm going to need 8 different inoculating loops because I'll be splitting each plate into 2 sections, and 2 times 4 is 8. I'll also need a Sharpie for labeling my plates. I'm going to need the set of conformation plates, which has 1 of each kind of plate that we poured. And finally, I'm going to need some E. coli 1 and E. coli 2 broth that I've prepared. That's my source of E. coli. So first, let's label the plates. Okay, as you can see, I have 1 of each kind of plate. So I have my LB, my LB amp, my LB strep, and my LB strep amp. So our conformation plates involve all 4 kinds. We're going to streak each kind of E. coli onto each kind of plate. My plates currently have a bit of moisture on them, so I'm just going to wipe them off with a paper towel because right now the Sharpie doesn't want to stick. I wiped each plate off and then I just drew a line down the middle as best I could. It doesn't have to be perfect. I didn't use a ruler or anything, but I'm just trying to split each plate in half. As always, we're doing this on the back of the plate. All labeling is always on the back of the plates. Now I've added a 1 and a 2 to each side. So that's just going to tell me where to streak my E. coli 1 and where to streak my E. coli 2. And for streaking, if you're working in teams of 2, I would recommend 1 person do E. coli 1 and 1 do E. coli 2. If you have a group of 4, then one of you could do each plate and do both strains of bacteria on it. You want to switch inoculating loops each and every time that you do your streaking. So this is going to be my loop for my first streak, and I'm going to do E. coli 1 first. So I make sure that my bottle says E. coli 1. I make sure I have bacteria in there, and the way to know is if it's cloudy. So this is a bottle on the left that I've never inoculated. This one I inoculated yesterday, nice and cloudy. When I open it, it will be stinky, and that tells me that I have bacteria. You don't lay the lid on the table. Be careful to avoid the sides of the container. Dip the loop in, swirl it around, pull it up and out. You should see that you have a loop full of broth. Then I take my plate, take the lid off. We want to do this fairly quickly. I hold my lid, and on the side that has a 1, I'm going to just streak across a bit. Then I turn the plate, streak a bit, turn, streak, and I'm done. And I tried to avoid crossing the line. Now I take my loop, put it in the plastic sleeve, and this goes into my waste bag. With a fresh loop, I'll be doing strain 2 onto the same plate. Double check that it's strain 2. Immediately cap your E. coli. Put the lid back on the Petri dish and the loop back in the sleeve. And now we're going to set this plate aside because this plate's done, and I'll do the next 3 plates. All 4 plates have now been streaked with E. coli 1 and 2, and you can see they're still inverted. As soon as you streak, you want to invert. We always culture our bacteria with our plates inverted and the agar side up, lid side down. Then you just restack your plates in any order. Okay, so these are my 4 confirmation plates. I've rewrapped them in tape, labeled them with my initials and the date that I streaked them. And these will now go in the incubator at 37 degrees for 24 hours, or I'll leave them at room temperature for 48 hours. And since I don't have an incubator, mine are going to be at room temp for 48 hours. So while we wait for the bacteria to grow on our confirmation plates, there are a couple of things. Number 1, your broth. I tend to get rid of all of my broth except for 2 bottles, 1 E. coli 1 and 1 E. coli 2, just in case we end up needing them. You shouldn't need them after this point, but I'm paranoid. So I would save 1 of each in the fridge, and the rest spray 10% bleach in there to kill them, and then discard them. The second thing is, make predictions about what's going to grow on each side of each plate. And you want to do that based on what you know about E. coli 1 and E. coli 2. Which one is resistant to which antibiotic is on each plate, and make sure you have those predictions before you view your results. Thanks so much for watching. The next video, which you'll see the link down below, is about what we're going to do next once we have our confirmation results. Have a great day.